Kia ora koutou katoa. My name is Yandiki. I'm going to be covering the last 6 weeks, or sorry, not 6 weeks, last 6 lectures in Biology 309. And I'm going to try and break it up a little bit and do some of them as shorter lectures because if we're going to go with these video lectures, which I kind of need to do because of the timing of the labs and the lectures, we may as well take advantage of the shorter format the videos allow. So for those of you who don't know me, I'm an ecosystem ecologist, I'm a microbial biologist, and I do a lot of ecological research on how fungi and other small and often unappreciated organisms actually control the world we live in. But for the purpose of this class, I'm going to talk about statistics and analysis of community data, which is something I do a lot. I'm not a I don't do statistics for fun, unlike some people, but I use them heavily, and I do program in R a lot. I've got a very much a programmatic focus in the way that I approach my statistics. So let's see if this will work here. Just to give credit where it's due, a lot of my work, a lot of my slides build heavily on work by Biol309, and I've got a link here in the slides. I'll also put a link on the Learn page to some of his resources, which you might find useful if you're looking for something to read up and understand what we're talking about a bit more. The data I'm going to use is partially from Vegan, some from my own work, and other collaborations I've been involved with. So I'll try to use a lot of real data in my lectures. And one of the types of data we deal with a lot are plant communities. So this is an example of a complicated thing to measure. You have to go out and you might measure in one plot the abundance of broom, flax, grasses, shrubs, even rock surfaces versus soil surfaces. You might also measure some environmental variables, carbon and nitrogen, phosphorus in the soil. And all of those things are what we call multivariate data, meaning that they've got more than one response variable that you might be interested in. And that's different than what you've looked at so far. In most statistics, you've been used to thinking about one response variable. Say, for example, pH in this example is controlled by either one or more other variables, so organic carbon or total nitrogen. Here, we're saying we've got a whole community. So we might say a community of birds, and we want to know how are they linked to soil nutrients, or are they? So now instead of having one response on the left side of the formula, we now have a whole matrix, a two-dimensional response variable. And again, we can have one or more predictors on the right side. So how do we deal with that? That is something new. First, we're going to talk about how do we even describe a community and how do we measure its properties. And that'll be the main topic for today. And then we'll move into some ways we can cluster and analyze community data. So I should be clear that when I say how do we do statistics on communities, I use the word communities to mean sort of any multivariate response. Although you might be thinking, I'm not an ecologist, I don't really care about communities, and that's fine. But you probably will deal with some sort of multivariate data, whether that's gene frequencies or what genes are upregulated or downregulated, whether it's profiles of enzymes with multiple responses or multispectral imaging, or even community responses to a poll. Almost all science at some stage gets into a multivariate data set. Okay, now that we're into R, let's take a look at some of the things we can do with this data. So we're going to be using the library Vegan, which is the Biol309's package, as I mentioned. Really tons of useful stuff there. And I'll just point out there, it has a function called Browse Vignettes Vegan, which if you run that, it will open your web browser and it will give you a link to some very helpful documentation, including on diversity analysis, on ordination and partitioning of variants, all of which are topics I'm hoping to cover during lectures. So we first have to get into the concept of a matrix. I'm going to try and do a short video on objects and what it means that R is an object-oriented language and what the sorts of objects are. But for now, let's just take it. There is a type of object called a matrix. It's a two-dimensional object, or rather, more accurately, it's a one-dimensional vector with also a long string of numbers that's associated with two dimensions, a number of rows and columns. But it's actually stored as this one long string. So we're going to load some data. This is the Barrow, Colorado Island data set. It's counts of trees in plots on a tropical island in the middle of the Panama Canal. And when we load it, we can use the function class to see that it's a data frame. Data frames are a type of list. It's a special type of list where each element of the list has exactly the same length. But I don't like working with data frames when we're really dealing with a matrix, so we're going to convert it to a matrix, and I'm going to call that BCIM, and just say, put the BCI data into there, but with the class as a matrix. And I'm also going to give it some row names. Right now it's just got numbers 1 through 50. I'm going to use the function paste to put the word plot and then that number together, separated by nothing. That's what the sep equals and then quotes means. So that it just makes it a little tidier, easier to see. So there we go. We've now assigned those row names, and I can look at the row names. Now the rows are all named plots 1 through 50. And actually, I can look at the matrix itself. Now, the matrix is very large, so I don't want to look at all the columns at once. Let's look at rows 1 through 4 and columns 1 through 10, just to start. There we go. And even that doesn't fit all on my tiny little screen. But you can see it's got different tree species, and they've got mostly zeros. But this species, for example, the first row is plot number 1. It's got 2 individuals of this Alcornaria costaricensis. Plot 4 has 18 of those, and so on. So different tree species with different abundances. Now, the fact that I've got this as a matrix gives me a whole range of tools I can use, and they're really important. So, for example, I can just ask how many trees are there in every plot, and that's just asking it to sum up the number of observations for each row, and that looks like that. And so we get the row sums being the count of observations, and we find that the plots have between about 340, I think is the lowest, to about 600 trees. So lots of trees packed in there. But the other cool thing I can do is I can convert the matrix from abundances into presence absences. That's a really important thing. It means that the number of trees of a species will no longer matter. I just want to know how many trees are present. So I can do that by taking this BCIM, my matrix, and asking whether it's greater than 0. And I'll just show you what that looks like just using the head just at the top first couple rows. It doesn't look the same, does it? It's changed from being numbers to being these what we call logical values, either true or false. Logical values are kind of neat. They've got some important properties, one of which is that being true is the same thing as being 1, and being false is the same thing as being 0. And so you can add them up, just like numbers. True, true, false is equal to 2, because 1 plus 1 plus 0. And so we can do math on them. So I could say, give me the sum of all the plots that have a tree, or of all the tree species that are in a plot, just as presence absence. So that would be doing the row sums again, but the row sums of that greater than 0 version of the matrix. So it's going to go along, and it's going to say, in this example, if we go to the top, we go 0, 0, 0, 0, 0, 0, 1, 0, so we're up to 1, 0, 0, oh, there's another one, so then I've got 2, and so on. It will just sum all those up. And when we do that, we find that if we were to keep doing that for all the trees, we would have 93 different tree species in plot number 1, 84 in plot number 2, and so on. So it's a measurement of the number of positive values in the matrix. By rows, you could also look at the columns of the matrix. So you could do call sums on the data, and that would be the number of individuals of each tree in the whole data set. Or you can take the matrix and again use that greater than 0 to convert it to a presence absence matrix and do the column sums on that. And this would tell you the frequency of each tree species. How many plots is that tree species found in? Think about that for just a minute because that's an important point. What it means then is I can sum that up. I can take the call sums, which will be the total number of tree species, and ask whether that's greater than 0. and then take the sum of that, and that will actually give me the total diversity. How many tree species are in my matrix? Now, as it turns out, this matrix doesn't have any columns that are all zeros. So it's actually those two, that number of 225 is also the number of columns. And we could add a line to our code. I will put all this code online for you, including anything I edit while talking. So we could ask, what are the dimensions of BCIM? And when we run that, we find that it's got 50 rows, which is the 50 plots, and 225 columns, which makes sense because that's the number of tree species in the data. Now, why am I going on about matrices? Matrices are a little bit different than other objects. They can be indexed just like a data frame. So you remember that BCI is the data frame version, and BCI.m is the matrix version. We could ask what's in the 4th row, 7th column of the data frame, and we'd get the value 18. And if we said BCIM, we'd actually get the same value, which makes sense because they're both, the one's just a copy of the other with a slightly different format. But you can also index both of these with a single value, without a comma. And what happens when you index a data frame with a single value? Actually, most people don't know. It's a weird thing to do. You wouldn't normally want to do it. But remember that a data frame is actually a list of objects that are all the same length. So it'll actually get you the 10th object in the data frame, which will be an entire vector. So this would actually give us the 10th species in the data set. Matrices do it differently. Matrices store every data point independently. So if I ask for the 10th point of the matrix version, I'll just, in this case, get a 1 because the 10th observation happened to have a value of 1. So why the difference is because when you save a data frame, it has a length of the number of columns in your data frame. When you save a matrix, it has a length of being the columns times the rows. Every once in a while, knowing that's really important. As I said, I'll try and do another little video on objects to explain more about it for those that want to know. But just if you get messed up at some stage, it can be a good trick to remember. Okay. What I want to do then is we have this matrix. It's huge. It's 225 columns. I can't visualize that very easily. So how can I see it better? And I'm going to show you 3 different plots that are sort of fundamental to visualizing data. 2 of them are widely used, and one of them is one that I personally just like to use, something I've developed a little bit. So we'll look at a species accumulation curve first. This is a really basic concept in ecology. It may not be as familiar to people in other fields, but we can do a function called spec_accum, which is for species accumulation, on our matrix. And I'm going to use the method called collector and then just plot it. So that's what this line of code is going to do. And when I do that and run that, it looks like this. What that's saying is we have 50 sites. If you took these sites in order from 1 through to 50 and said how many species I found, if you just took the very first site, this is telling us it must be somewhere just under 100 species within that plot, and we could test that at the row sums of the first plot, and we see it's 93, and in fact that fits with the collector's curve perfectly. The other thing you notice with the collector's curve is it's not a smooth line. It's based on the actual order of the plots that we took them in, which could be useful if they were in some sort of logical order. We can also look at the curve smoothed out, and so this would be doing it without the method collector. So this is actually, if you look at this would actually be the method exact, because that's the default method, which you can see by looking at the arguments when you pull up the function. So what this does is it's just like a collector's curve, except it's randomizing the order of plots and then giving us the average value and an error measurement around that value. And what you can see then is it smooths it out, and we find that on average, we get, again, something just under 100. And that would actually be the mean of the row sums of the presence-absence matrix. So if you look at that, it's about 90.78 would be where that line starts. But both the collector's curve and the species accumulation curve are going to end at the same spot. They're always going to end at 225 species because that's the number that are in our data set. Just to give you an example from some real data I have, this is a study we did of 5 different land uses across New Zealand. You're going to do a similar data set, slightly different collection of plots in lab. But here we look at 5 different land uses: natural forest, planted forest, low-producing grassland, high-producing grassland, and perennial cropland, in usually vineyards. And we looked at the species accumulation curves using DNA. And in this case, we've got fungal DNA I'm showing you, 15 plots. And what you can see is that the natural forest always had higher diversity. The planted forest, which is the dark green line, had the lowest diversity. And you've got everything in between. And you can right away from this read not only the difference among the different land uses, but also their average species richness per plot, because that's what you get the left-hand side, and their total species richness on the right-hand side of the plot, and look at those differences. You might also notice the lines of the bottom 2 lines look like they're about to cross. And this is an important point. If you looked at these curves and I asked you which is higher diversity, planted forest or perennial cropland in New Zealand, you'd have a really hard time saying, because there's just a small difference, and it looks like maybe if I took another 5 or 10 or 20 more plots, you'd actually find more species in the planted forest than we did in the perennial cropland. So that's going to be the next thing we get into in a couple slides, is how do we actually extrapolate these values up. But let's go back to R. So that was one very useful plot. So that's the species accumulation or collector's curve plots. The other thing we can do is look at a rank abundance diagram. This works by looking at the call sums of the matrix. So that is the abundance of each species, and then sorting them in order. So that's what this part of the equation does here. And then I'm just going to plot those in the order that they come. So it's their abundances in the order from the most abundant to the least abundant. So I'm using the function sort to get them in the right order. This type equals b gives me a graph with both points and lines, and I want to log the y-axis. It's convention that you show a rank abundance diagram with a log transformed y-axis. And when I run that bit of code, you get this curve to the data that shows us that we've got some species that are very highly abundant, a lot of species at intermediate abundances, and a small number at the end, this tail of species that occur only once in the data set. Those species abundance distributions, or SADs, they sometimes get called, are really informative about the structure of the community, and there are books written about just this one diagram and how you can interpret it to understand your data. You can also do the same thing. This first version I gave you was based on the total abundance matrix, but I could also do it based on frequencies. So again, I've got 50 plots, so I could give it a maximum value of 50, meaning a species was found in all 50 plots, and not pay attention to how abundant it was within that plot. So I just, again, convert the matrix by using this greater than 0 conversion. And I get quite a different curve. So you'd find that the frequency abundance graph here is maybe a little bit different and gives us some different information about the structure of the community. The last graph I just want to show you, and this is one that I like to do, it sort of puts those 2 previous ones together. What it does is looks at the abundance of a species when it's present. So that is, if a species is in a plot, how abundant is it as a proportion of the total? And it does that as a function of how frequent is it across all the plots. That gives you a measurement of abundance that's independent of its frequency. So a species can be highly abundant at one plot and absent elsewhere. It'll still show as a high value here. And when we do this line of code, here I'll break that up on the code so it's a little easier to see. So we've got the call sums divided by the presence-absence call sums. That gives me the corrected abundance as a frequency, and then as a function of frequency. And what you can see is this interesting curve here. And I'm just going to tidy that up a little bit. Let's just fix this for the smaller window. So this just labels it properly. It's always good to have your graphs labeled properly. So I'm same basic start, log both axes, but now I'm labeling the axes as frequency and as average abundance when present. I'm also taking away the box because that wasn't adding any value to the figure, making the points into these solid points and orienting the labels horizontally so they're easy to read. And then that's what this graph looks like. So in this case, what it's saying is there are a bunch of species that are found in 50 plots that are often very abundant, but there are also some pretty widespread species that aren't that abundant. And there are a few species that are found in only 1 or 2 plots, and they tend to be relatively low abundance compared to the other tree species. But here's one that's actually quite abundant in the 1 plot where it's found. So you've got maybe 9 individuals in that 1 plot. And again, just to show you that this can be useful in real data, in data from New Zealand, my own data. This is a plot of fungal data looking at dominance, that is again the abundance when present as a function of occupancy for 2 different groups of fungi in deadwood on the west coast. I'm going to talk about this data at some point, but it's a much larger data set. And we can see that, say, basidiomycetes, shown in this yellow color, tend to be both widespread and high abundance. But ascomycetes can be very abundant, they're just not very widespread, and we could start coming up with some good ecological explanations for that. But at this point, just for the stats course, I just want to show that this is a useful way to examine community data. So from there, I want to come back to this question of how do we determine how diverse a plot is? And remember that those curves, when we do a species accumulation curve, can cross, so we have to think about that. We also have to think about what does diversity really mean. So if I show you these 2 communities, one on the left, which has 8 dogs, a fox, a koala bear, and a frog, and then I show you this one on the right, which has 3 dogs, 3 koala bears, 3 frogs, and 3 foxes, which of those is more diverse? And obviously the answer to that question depends on how do you measure diversity? What do you consider to be a measurement of diversity? So on the one hand, we might say that it's just the number of species. So this is what we call species richness. It is simply the number of species in a plot. And I've already shown you one way to do that using this row sums of a matrix that's greater than 0. So we can do that. There are other ways we could calculate it, though. One thing we could do is write a function. So if you consider that there's a formula for species richness, it would be the sum of the proportions of all the species raised to the 0th power. The reason we use the 0th power is that it converts any number, any value, into a 1. So it's literally just converting the proportions into a presence-absence matrix. So if we write that as a function, we would take a vector x, and we would say, first of all, get rid of any values of that x that are less than or that are 0 or less than 0. So that cleans up all the absences. Now we just have a vector of positive occurrences. We can convert that into proportions by dividing it by its own sum. And then if we take the sum of that matrix raised to the 0th power, it's just a count. It's literally saying how long is that. And so we could take that function and we can now, remember the function is designed for a vector. We've got a matrix. So it's designed to take a single row of values and we're giving it 2 dimensions. So we have to use this function called apply. What apply does is it says, take this matrix, bci.m, on the first margin, that is by rows, so we're giving this a value of 1. If we wanted to do something by columns, we'd give it a value of 2. But because we want it by rows, we're going to say on the first margin, apply this function richness f that we've just written. And if I go back to R, so we can do that, here we've got, there's the original way we had of calculating richness. there's my function where I remove any zero. This one's better commented than on the slide. I remove any zero values. I convert it to proportions, which really has no point. It doesn't do anything in this function, but you'll see why I'm doing it in a few minutes. And then I can take the sum of those raised to the 0th power, apply that to my matrix, and when I do that, and then compare the values, so I put the values into richness calc, and I compare them to this other way of measuring richness that I showed you a second ago, it tells me that actually it gives me the same exact number. And it turns out there's a third way I can do it, which is a built-in function in vegan called spec number, or species number, which again I can just run on the matrix, and again compare it to my row sums version, which I called richness, and again every value is exactly the same, so it returns all true when I do a direct comparison. So 3 ways to calculate richness. You can take the sum of the rows that are greater than 0 after converting the matrix into a greater than 0. You can write a function that takes the proportions of each species and raises them to the 0th power and then sums up those. Or you can use the built-in function spec number, and they all come to exactly the same value. That's one measurement of diversity. If we go back to our slides, by that measurement of diversity, these two communities would have exactly the same diversity because they both have the same number of species. But some of you might be looking at these two communities and thinking, no, clearly the community on the right has a higher diversity because it's not all dogs. The one on the left has 4 species, but it's mostly dogs. And so that gets us into a really interesting measurement of diversity. This is called Shannon diversity. It also gets called Shannon-Weaver or Shannon-Weiner, but it's a measurement of entropy. And it isn't just from ecology. This is a measurement of the compressibility of data that is universal across all sorts of sciences. It actually comes initially from trying to code messages and asking how well can you predict the next letter in a series of letters. It has applications in genetics, it has applications in all sorts of fields, and it is actually very much linked to the concept of entropy. So one way to define it is, on average, how many times would I have to ask a yes-no question to identify the next species if I were to draw them out of a community? So you can imagine that first community, if I said, is it a dog? That very, very often I wouldn't have to ask any more questions. You say, yes, it's a dog. And I pull out the next one and say, is it a dog? Yep, it's a dog again. Is it a dog? No. Is it a frog? No. Is it a koala? Yes. So in that case, I had to ask 3. But the average number will be fairly small. So the way we calculate Shannon diversity is we take the proportions, we multiply them by the log of the proportion. So each proportion times its own log, and then we sum those up and times it by -1. So it's a strange little formula, but easy enough to calculate. So just like we did for richness, we can calculate that as a function. So I'm going to write this Shannon F function, where I'm going to take the values of x and get rid of those that are zeros. I'm then going to take each one and convert it into proportion, and then I'm going to take the negative sum of each value times its log, remembering that in R a log is, a natural log is just the function log. And then we can again use the apply function on our matrix to put that onto each row, and then we put in our own function we just wrote to get the Shannon diversity. Now let's go back to R again. So I've got that here. There's the function, just as I just went through it with you. We can then apply it, and then we can look at the Shannon diversities, and we get this series of numbers. Now you notice those numbers are very different than the number of species that we had before. For species, we had numbers, well, we can look at them real quick. We had numbers like 93 up to 105-ish. But for Shannon, we're looking at these sort of strange numbers of 3 to 4. So we need to think about what do those really mean. We can also, again, there's more than one way to calculate this. we've written a formula because I want you guys to be familiar with the idea of writing your own formula. But there is also a built-in function in vegan that can do this for us. It's just the default diversity function. So if we say, what's the diversity of BCI, and then compare that to Shannon, to our calculated version with our formula, what we find is that they are exactly the same values. The last way I can do it is a slightly different version, which is something called Simpson diversity. Simpson diversity is a measurement of diversity that's most sensitive to abundant species. So it just takes the proportions of each species and squares them, and then sums those up. And then you subtract that from 1 to actually get the Simpson diversity most people do that way to get it to have a larger value when you've got higher diversity. So we can do that again simply by using a built-in function. We can use the diversity function again with the index being Simpson rather than Shannon. And we can go ahead back to R and try that. So there's the built-in Simpson function, and I can plot that as a function of richness. And you might see that the Simpson diversities are all pretty high, except for this one value that's quite low. And they're not that well correlated with the total number of species. There's a little bit of a correlation. So 2 different measurements of diversity giving us quite different values. We can also look at writing our own function. So just like we did for richness and for Shannon, we can write a function that takes the values, so it takes out anything that's 0, converts them to proportions, and then just sums up those proportions squared and subtracts it from 1. And then we can apply that to our matrix and compare it to what vegan calculates with the built-in function. And again, we find that our formula is just as good as the built-in function. I do want to note that there is that one outlier, whichever way we calculate it, that one with about 83 species for richness, but a Simpson diversity of around 80. The fact that it's low diversity might tell you that there's one species that's making up a lot of the diversity in that plot. And in fact, we can look at it, and this is actually looking at that one plot where the Simpson diversity is below 85. We can then sort the abundances, and what we find is this one tree, Gustavia superba, there's 247 stems of that one tree species in this plot. The next most abundant only has 93. You can imagine how boring that must have been to measure. That it was like, oh, it's another Gustavia superba. They spent all day sitting there measuring the same tree species, and that's why it's coming out as being a low Simpson diversity. Now, that gets us into a problem. If we look at diversity, we need to think about these metrics, these values. 0.8. What does 0.85 to 1 for Simpson really mean, and what does this 3 point something mean for Shannon? And we want to come up. We might have some desires for how those values should behave. So one of the things we might want them to do is, if we combined 2 communities that had shared no species in common, we might expect that the diversity would double. So we can look at that. So if we had 8 species, and let's just make them all exactly equal, so we've got one individual of each of 8 different species, each one would make up 12.5% of the community. And then we could have a 16 species community, so add another 8 species with no overlap. So I've added to these species, I've added a unicorn and a gorilla and a rhinoceros, so there's no species in common. So now each species makes up 6.25%. It would seem logical that the second community should have twice the level of diversity of the first community. So let's see what actually happens in R. I'll first create those 2 communities. So I've just created a community of 8 species. I give them each abundances of 0.125, and a community of 16 species, again, just taking 1 and dividing it by 16 to get the abundances. And if I looked at the richness, so remember, all I have to do is take the sum of those values that's greater than 0, I would find that the community with 8 species has a richness of 8, and the one with 16 species has a richness of 16, which is good. That's exactly what we should be getting. But if I looked at Shannon diversity, for example, the Shannon diversity of the first one is 2.08. And when I double the number of species, the Shannon diversity is 2.77. So it's not twice as diverse measured by Shannon diversity. And same thing when I do Simpson diversity. If I have the community of 8, it's 0.875, and when I have a community of 16, it's 0.937. In fact, the closer I get to 1, the smaller the change I get by doubling the number of species, because it can't go above 1 for Simpsons. So that's a problem. It doesn't make sense to have a measurement of diversity that when you double the number of species, and they're all still at the same relative abundances, they're all equally abundant, you would expect the number of species to, or the diversity measurement also to double. This has led us to coming up with something called Hill numbers, which have been popularized by an ecologist by the name of Jost. And these are ways of converting Shannon and Simpson and other diversity metrics to what we call effective species numbers. One way to think about this is, say you had a balloon. If you blew 1 liter of air into that balloon, the volume of the balloon would be 1 liter. And if you blew another liter in, the volume would be 2 liters. But you wouldn't expect that the radius or the circumference would necessarily double. However, if you were clever and you knew what the formula was for volume as a function of the radius or the circumference, you could calculate back a value that would double, which would be the volume. So this is kind of the same idea. We've got 3 different measurements of a community: richness, Shannon, and diversity, but they're all measuring some different aspect of it. What we want to do is put them all back onto the same metric, so that if a community had exactly equal proportions, they should all give exactly the same result. And the way we can do that for Shannon is if we just take it turns out that if we just take the exponent of the Shannon diversity, that behaves nicely. So I can just show that, going back to R. If we take the exponent of Shannon for the community with 8, we get 8 species. And if we take the exponent of Shannon diversity for the 16, we get 16 species. So you can convert from Shannon back up. And there's also a conversion for Simpson diversity, which is to take 1 minus the diversity and then take it as a reciprocal. And again, you see that you can recreate the number of species from that value. So those are what we call effective diversity. They are a measurement of diversity, but in the case of richness, it's based simply on presence-absence. In the case of Shannon, it's a measurement of the number of species, but weighted by their proportion in the community. So Shannon will have the same effective diversity as richness if every species is equally abundant. And the more they get away from equal abundance, the lower the Shannon effective diversity will be. It will always be lower than richness. And Simpson, the same thing, but it's even more sensitive to those abundant species in your matrix. There are formulas for each of these that allow you to convert from the standard measurement back to an effective diversity. But this gets into the idea that maybe there's one universal formula, and it turns out there is. This is the universal formula from which you can calculate all of these other indices, and it's called Hill numbers, where the Hill number is the Q in this equation. The diversity for Q equals 0, this reduces to being the number of species in your community. If Q becomes 1 and you plot it into this equation, it actually doesn't work. It actually isn't defined for Q equals 1. But if you take the limit of the equation as it approaches 1, it ends up being the exponent of Shannon diversity. So you've managed to recreate Shannon diversity. And if you put 2 in the place of Q, it ends up being the Simpson diversity. So this has led to an idea that all of these diversity metrics can be seen as a continuum of Hill numbers, with 0 being richness, 1 being Shannon diversity, and 2 being Simpson diversity. But you could go on up to infinity, looking at different metrics of diversity, and it's becoming commonplace to look at a profile of diversity across these different metrics. So This just sums that up. Hill numbers provide a continuous scale of diversity metrics. They give us the effective diversity, so the numbers are no longer these kind of meaningless 0.87s, 3.2s. They're actually, they can always be interpreted as the number of species if all species abundances were equal. And the nice thing about working with effective diversities and Hill numbers is that they have the doubling property, which is that if you double the number of species, the index of effective diversity will also double. So that's a really important property of that metric. Now that's not the end-all and be-all of diversity. There are other metrics, and I'm not going to talk about phylogenetic diversity, but I just point out that there are also other aspects of diversity. For example, if we were to look at these two communities, both of them have 8 species. But if you were to look at it, you would probably say the one on the left clearly has a higher diversity because it's got all sorts of different phyla of life within it, whereas the community at the right, they're all kind of related to each other. They're all mammals. And if we were to put them on a phylogenetic tree, you would see that this first community here has a relatively large phylogenetic diversity, whereas the second community, despite having the same number of species, has a much lower phylogenetic diversity. But that's going beyond where I want to go with this course. So we'll introduce the concept of phylogenetic diversity, but we won't actually do any exercises with it. Now, I raised the question earlier of those collectors curves or species accumulation curves that looked like they might cross. So this is the idea of once you've measured diversity, how do you compare it across different treatments? And remember that the numbers we have so far are based on observational effort. And if you put more effort into observing something, you'll see more species. So if you're comparing 2 plots and you spend 5 hours measuring all the plants in one plot, and then you go to another plot and you just spend 5 minutes looking at the plants, you can guess that the one where you spend 5 hours, you're going to find a lot more species. So we want to find a way to compare diversity when you have that different sampling effort. And this is just the example of that. Here I've got 15 plots that I've measured, but you can see none of these curves have actually plateaued. They're still going up, and so I can't really predict where they're going to end up. When you've got this problem of unequal sampling effort, that's where we get into the idea of using something called rarefaction. Rarefaction has a couple different meanings, but for right now, we're just going to use it to mean sampling all of your plots at the lowest level of effort. So because you can't, in this case, we've got a red plot and a blue plot, it looks like the blue plot has fewer species, but really to compare it to the red plot, we have to sample an equivalent number of individuals. And we can do that by rarefaction. So rarefaction estimates species richness when you have the same depth or same effort of sampling, and it inherently throws away some of your data. It's based on the smallest data set you have, and then throwing away the data from the bigger data set until it's the same size. We can take an example of that. So if we look at this BCI matrix, we know that there are different number of trees in the different plots. And maybe we want a measurement of diversity of trees that's not biased by the number of trees. We just want to say, if there were the same number of individuals, would this plot have higher or lower diversity? So the first thing I want to know is what's the fewest trees in any plot? And if we do that, we get this value observations min. So I'm just looking at the minimum of the row sums of the matrix. And when I look at that, I find my smallest plot only had 340 trees observed in it. So I can then use this function rarefy to look at my matrix, and I have to tell it how many things to sample. Actually, I'll show you what happens if I don't do that. So if we run it without giving it a minimum number of observations, it'll give me an error, but it gives me an informative error. It'll say, the size of sample must be given, hint, smallest site maximum 340, which is exactly what I got for observations min. So then I can run it and give it the sample size of observations min. And if I look at that as a value, we can just go srar. It gives me these equivalent sampling efforts for all of my plots. So how many species would I find if I sampled the same number of trees from every plot? And we could plot that as against richness. So in this case, I'm plotting the observed number of species, that is the raw richness, versus the rarefied number of species. And you can see they're relatively well correlated, but there are some outliers that have either higher or lower diversity once you rarefy than you would expect. And that implies some bias by the number of species observed. We put a line on there, and you can see that they always fall, the rarefied species will always fall below that line, except for at least one value that will fall on the line, which is because you used the smallest sample size. So obviously its rarefied diversity and its observed diversity are going to be exactly the same value. You can also use this function rarecurve. Rarecurve in this case is kind of a mess, but what it's doing is looking at as you increase sample size, how does the number of observed species increase? And then this vertical line is that min value we use of 300, I think it was 340. And then it's drawn a line back to the y-axis right where each of those blue curves crosses the line. So that's one method of comparing diversity across samples when you have unequal sampling effort. The other thing you can do is look at extrapolation. So you can't just fit a curve, unfortunately, but you might say, where am I going? If I was to extend out, what would the real diversity be? If I could really sample every last thing in this world, if I could sample every vineyard in New Zealand, how many species would I find? And obviously that's an appealing number to have. It's like, wow, what's the true total diversity? I don't want to just rarefy and say, oh, if I sample 5 vineyards, how many species will I get on average? I want to know if I sampled every vineyard, how many species would I get? Just as an example. So how do we extrapolate outwards? You can't, unfortunately, just fit a curve to the data because the shape of the curve is very hard to model. And just fitting a curve doesn't really consider rarity very well. The shape of the curve might be different out at the far end when it's being driven mostly by picking up really rare species. In fact, that's kind of a truism here, is that when you are extrapolating, you're really asking about those rare species. How frequently are you going to pick up something that after 150 plots you've never seen before, and yet you pick up one more species? So most of our information is now not in the common things, but in those rare things. So we do this through a function called, well, there are various functions, but I'll show you one called chow. This is the chow estimator. It takes the number of observed species, which I'm just calling S-obs, and then it takes the number of what we call singletons and doubletons. That is, the species with a frequency of 1 and species with a frequency of 2. And in this case, it just takes the number of observed species and adds to it the number of singletons squared divided by 2 times the number of doubletons. If the number of doubletons is 0, there's a slightly different formula you can use as well. And so we can do that. Again, it's built into R in the spec pool function. We'll go back to R. So if I take, these are our counts, so they're just the frequency of every species in my data. And I can just ask, what is the sum of the counts that are greater than 0? That's my species observed, plus the number of them that are found once, and then square that divided by 2 times the number of them that are found twice, remembering that this counts equals 1 is going to be true or false, and I can sum that up to get the number that are true. And when I run that, it's going to come to a value that there are, if I was to extrapolate this data out, there are roughly 239 tree species that I would eventually find. Vegan uses a very slightly different formula. I'm not going to go into why it is, but if you check it against vegan, it runs all these different formulas, and chow is the first one it runs, and that comes up with 236, so very close to the value that I got. Calculating the standard error of these is quite challenging mathematically. I'm not going to show you how to do it, but vegan does it for us, so we can also put a standard error on that measurement. And then it shows us a few other functions, the jackknife and the bootstrap, two different jackknife functions on the bootstrap, again with variances associated. So these are just some of those other measurements. So this is the abundance-based estimator. Again, it's looking at the number of infrequent and frequent species, takes into account more of the different levels of abundance, but it's still highly influenced by the number of rare species. The jackknife is based on removing species that occur once or including doubletons in that calculation, but again, it's driven very much by the number of species found once. And so you end up with these different values. And I'll just run through this in R then. If we were to load some data, so I'm going to take in some environmental data, and I'll just show you that really quick. This is also from the Barrow, Colorado Island. We can see, do that better. We can see that for each plot, I've got some coordinates, precipitation, elevation, age category, geology, habitat, stream, whether there's a stream in the plot, and the environmental heterogeneity. And I might think, just type that in so you've got it in your version of the code. I might think, well, maybe plots with a stream in them will have a higher diversity than plots without a stream in them, because I might think that maybe the streams are going to lead to being more heterogeneous environments and a better spot for trees to grow. So I could run this species pool on my matrix, but in this case I'm going to give it these pools. I'm going to say compare the plots that have a stream with those that don't have a stream. And when I do that, I find for chow, for example, those that don't have a stream have an extrapolated richness of 226 species, and those that do have an extrapolated richness of 195. So actually it doesn't support my hypothesis. In fact, when I extrapolate the diversity, it looks like those without a stream have a higher diversity than those without. But I do note the variances on those are reasonably high, so I'd need to do some statistics to actually test whether that's significant. And then we can plot those with species accumulation curves. And actually what you find is that on the species accumulation curves, they're really very, very similar. So if we were to rarefy, we'd find no difference. And when we extrapolate, we do find a little bit of a difference, which might lead you to question whether you really want to extrapolate or not, because it is very much based on a small data set and you're taking that up beyond the limits of your data. Chow estimators are pretty good. If you were to take a large number of subsamples, the number of observed species is in this dashed line here, the darker dashed line. And what you can see is it does start to come up, but it comes up slowly. The nice thing with a chow estimator is it reaches the asymptote more quickly, and then it's pretty much flat. I'm not so fond of the jackknife and bootstrap values because they don't really asymptote. They just increase more quickly than your observed values. So again, observed values are in the dashed line. The first order jackknife is here. Bootstrap, same thing. And so they're not actually, they're just giving you a bigger number, but not any more robust of a measurement of where the line might be expected to asymptote if you did take a big enough sample. One key point here is that for both of these, you still need a pretty big sample. For you to be confident, even in the chow estimator, which is one of the best at asymptoting, it first crosses the asymptote at around 30 samples. So these are not techniques that allow you to do a poor sampling of your community. They're just techniques that once you have a reasonably good sample, allow you to extrapolate upwards. The value should asymptote. If the estimator of your extrapolated diversity, this is you're trying to estimate where that curve will asymptote, if that fails to asymptote, then you're no better off. You've just come up with a different number, not a more robust number. So use these with caution to try to extrapolate diversity, but remember you're still limited by the size of your sample. That pretty much covers what I want to talk about with diversity, and that's about half of the first lab. We're going to cover clustering in the next lecture, but you can probably make it through the lab. I've written the lab instructions in such a way that you should be able to do clustering without actually having seen the lecture on it yet. I've just made that a little more explicit. So hopefully that's clear. I will put up a short video on objects as well for those of you who want to better understand what are the different types of objects in R and why are they important. Thank you for listening, and I will talk to you guys later.